everyone. It's 1 p.m. Eastern Time. We're going to get started with our webinar today. I'd like to start by thank you for joining us for our first in our international webinar series of 2013. This one is regarding high-quality microplates for immunology assays. My name is Mackenzie Ferrone, and I will serve as your host for this webinar session. Over the course of the next 40 minutes, Dr. Ulrika Hanisch will present a brief overview of ELISA methods, microplate features for immunology assays, surfaces for passive absorption and covalent binding. If you have any difficulties with your audio during the webinar, we would like to remind you to please make sure you are listening directly through your computer speakers and the volume is turned up to a reasonable level. If you continue to have difficulties, please use your chat feature to message the host, and we will do our best to get the issue resolved. Please feel free to use the WebEx chat feature as difficult to ask questions. You can submit your question at any time, and we will make a sincere effort to respond to all before the end of the webinar. We are so happy to have one of our own speaking with you today. It's my pleasure to introduce Dr. Ulrika Hanisch, who is a product manager in the Greiner Bio-One's Research and Development Department. Dr. Hanisch holds a PhD in biology from the Institute of Microbiology at the University of Karlsruhe in Germany, involved with the development of ELISA and protein crystallization microplates for the past 10 years. I will turn it over to Ulrika.
Thank you very much, Mackenzie, for this kind introduction. And welcome to everybody as well from my side to today's webinar with the title High Quality Microplates for Immunological Assays. I start with a short introduction about phase assays in microplates and switch to the main focus of this webinar, properties and quality aspects of microplates used for immunoassays. I will end my presentation with some remarks about critical parameters for assay development. So let's start with the introduction. Immunoassays are common tools in laboratory medicine. They are used for diagnostics and disease monitoring, environmental and food analytics, as well as doping tests. And they are a powerful tool in basic science. ELISAs are a rapid, sensitive, and highly reproducible method which can be used in a high throughput manner. Assays conducted in microplates are multifaceted. However, the principle of all assay formats is the selective interaction of an antigen and a specific antibody. The surface of the walls of the microplate acts as a solid surface where one component, the antigen or the antibody, is immobilized by passive absorption. Molecules not specifically interacting with the immobilized component are away during the assay. But both components to the microplate surface is made visible by enzyme activity, direct labeling. One way to classify ELISAs is to divide them into direct and indirect assays. In a direct assay, one component, in this example the antigen, is immobilized on the microplate surface. It interacts with a second component. Direct ELISA, neither the antigen nor the specific antibody are labeled. For detection step, an unspecific secondary antibody enzyme conjugate is used. A common ELISA format where the aim of an assay is to detect an analyte is the so-called sandwich ELISA. In a sandwich ELISA, a specific capture antibody is mobilized on the microplate surface. In a second step, the antigen contained in the sample binds selectively to the capture antibody. Second specific antibody is added in a subsequent step and interacts with a different epitope of the antigen. In the last step, an unspecific secondary antibody enzyme conjugate is used for detection. Prerequisites for a sandwich ELISA are availability of two specific antibodies of two accessible epitopes on the analyte. Competitive ELISA, an unknown amount of analyte in the sample and an introduced concentration of ad-labeled analyte for binding to a specific capture antibody adsorbed to the microplate surface. No analyte is contained in the sample. Only labeled analyte will bind to the capture antibody, which will result in a high signal intensity. More analyte is contained in the sample, less labeled analyte will be able to bind to the capture antibodies, which will result in a weaker signal. Slide 9 is a short summary of possible modes of detection. Horseradish peroxidase and alkaline phosphatase are the most widely used enzyme conjugates. They combined with a colorimetric substrate in a classical ELISA, or higher sensitivity is needed with a luminescent substrate in a luminescence immunoassay. The most common method in the meantime for direct labeling is the usage of a fluorescent dye like FITS, which substituted radioactive labels in the meantime to a large extent. Let's move after this short theoretical introduction to the topic of microplates as solid surface. If you're looking for a fitting plate for a new project, you have to make your choice out of a variety of microplates. The name of this webinar is to make this choice easier by pointing out the main features of microplates. ELISA applications, these are format, color, surface properties, and last but not least, constant quality. Immunoassays are available from the 96-well up to the 1536-well format. Critical aspects for the choice of format are the number of samples to be processed and the technical equipment available in your lab. Mostly 96-well microplates are used. They're available in the standard format as well as with a half-area geometry or as strip plates. Area design is an interesting alternative to save sample volume without changing to the 384-well format. Because of their well-designed half-area plates, allow a reduction of sample volume up to 45%. The path length of 0.5 centimeters, a volume of only 80 microliters in half-area microplate instead of 175 microliters in a standard plate is required. In addition, half-area microplates are compatible with most standard devices without any special adaptations, and manual handling is more comfortable than with 384-well microplates. Especially for the usage in diagnostic kits, it is advantageous to have the opportunity to adjust the number of wells to be used at once to the number of samples to be tested. To fill this demand, strip plates are available in a wide variety. Break strip plates allow to process single wells, whereas new bottom strip plates feature the separate handling of strips of 8, 12, or 16 wells per strip. ELISA microplates are offered with different well bottom designs. U-plates guarantee easy and residue-free pipetting and washing, but are, due to optical properties of the round bottom, only recommended for plus-minus analysis and not for quantification. Bottom plates are perfect for precise pipetting, but as well only a plus-minus analysis. Bottom microplates, in contrary, offer excellent optical properties and are very well suitable for precise optical measurements. The design combines the perfect optical properties of a flat bottom plate with rounded corners for residue-free washing. An important plate feature is the color of the microplate. MON stands for transparent ELISA plates for colorimetric measurements. Fluorescence detection, usually black fluid drug plates are used, whereas luminescence assays are in white lumidrag plates. Pigmentations, black and white, help to overcome the drawbacks linked to fluorescence and luminescence-based assays, which are a high background, autofluorescence, and well-to-well crosstalk. Very important plate feature when it comes to assays based on immobilization is the surface property of a microplate. Polystyrene is the most widely used material for immunological microplates, highly transparent and therefore ideally suitable for optical measurements. Polystyrene is by nature a relatively hydrophobic material. However, its properties can be modified by a physical treatment to introduce hydrophilic groups to the plate surface. Hydrophilic groups are beneficial for the binding of polar biomolecules and biomolecules with polar regions. This graph here shows a comparison of the oxygen content of a non-treated polystyrene surface and a hydrophilized high-binding surface. The oxygen content of the non-treated surface is about 0.5%, whereas the hydrophilized high-binding surface contains approximately 2.5% oxygen. The number of hydrophilic groups on the surface can impact the binding capacity of the plate. When this graph is a comparison of a high and medium binding plate in a fluorescence immunoassay, where IgG is immobilized on the plate surface and detected with a FITS labeled antibody. Binding capacity for IgG is significantly higher on the high binding in blue than a medium binding surface in red. However, behavior does not only depend on the surface properties of the microplate, but naturally as well on the properties of the molecule to be immobilized. This graph here shows the binding curves of a set of biotinylated proteins: 5.8 kilodalton peptide hormone insulin, albumin, IgG, and protein A, detected with Streptavidin horseradish peroxidase conjugate. So all molecules have been immobilized on the high bind surface. The curves, due to the different molecule properties, are significantly different. For cell-based immunoassays, most often microplates with a tissue culture surface are recommended. Therefore, we are often asked if this kind of surface can be used as well for non-cell-based ELISAs. Some light on this question, we compare the performance of a high binding and a tissue culture plate with two assays: an indirect ELISA, where insulin is immobilized on the plate surface, and a direct human IgG-based ELISA. Both IgG and insulin bound very well on the high binding surface. However, insulin did not bind significantly to the tissue culture surface. Even the binding of IgG was much weaker. It's likely the high charge density on the tissue culture surface, which is beneficial for cell adherence, is not beneficial for protein absorption in an ELISA, rather prevents binding by rejection. We definitely do not recommend tissue culture microplates for non-cell-based ELISA. Very important for the performance of immunoassays, besides microplate properties, is a constant quality of the microplates used. To achieve this goal, use at Greiner Bio-One only high-quality charge material for the production of our microplates. The production process is well established and monitored, and the performance of the ELISA plates is controlled by biological testing. Each material batch comes with a specification of the raw material supplier and is additionally controlled by our spectroscopy, by our QA department for confirmation of the raw material type and exclude impurities. The tested raw material batch is then used for a sample production of 100 microplates. 5 out of 100 plates are subject of immunological testing against a reference plate. Our quality criteria are the following: interplate coefficient of variation with a benchmark for phase homogeneity has less than 5% for colorimetric and 10% for luminescence and fluorescence detection. Our plate CV has to be less than 10% and sure of the optical values of all tested plates to be within 100 plus minus 10%. If all criteria are met, the raw material batch is released for production. Injection molding parameters for production are determined for each mold in a validation procedure, stored permanently. During production, these parameters are controlled electronically. Dimensions and blank values are subject to an in-process control procedure during production. Our ELISA plates are controlled automatically by an integrated camera system. The system is doing a 100% control to detect, for example, scratches, floor marks, air bubbles, or incomplete plates. After plate assembly, a robotic arm introduces the plate into the camera system. On the picture, you can have a look inside the system. The plate is illuminated and recorded. The recent picture is shown on a screen, and if the system detects a default, the respective plate is automatically eliminated. The result of this course of action is very consistent microplate quality. This here shows the results of an IgG ELISA conducted in high binding microplates out of 10 lots collected over a time period of 4 years. So of the optical density values for all tested plates is 94.3%, and the C for all tested plates is well below the 5% quality criterion. Product transparency and traceability, the information about the raw material batch number, and the PC, the input control, are included on the box label of our immunological microplates. In the box is a batch-related certificate of quality, adding raw material control, sample inspection, binding properties, and the absence of detectable DNAs, RNAs, human DNA, and endotoxins. Evaluation of raw material batches or production lots is possible according to prior agreement and frame conditions. For some applications, adsorptive binding to a physically modified polystyrene surface, like high and medium binding surfaces, is not feasible. Alternative is to take advantage of the strong non-covalent interaction between Streptavidin and biotin. Here, Streptavidin-coated microplates act as a surface on which biotinylated molecules can be attached very effectively and detected with appropriate antibodies. Coated microplates are available in the 96 and the 384 format. For some applications, when adsorptive binding does not work at all, or the signal intensity on physically treated plates is too weak, or when unspecific background signals are too high, Greiner Bio-One offers microplates with a 3D epoxy surface for the covalent binding of biomolecules, proteins, peptides, or nucleic acids. How does this work? A 3D epoxy surface is a stable chemical modification containing epoxy groups. Epoxy groups react spontaneously with nucleophilic groups like thiols, amines, or alkyls to stable covalent bond. 3D means that the chemical modification of the microplate surface results in a three-dimensional functional matrix with a layer thickness up to 50 nanometers. Derivative epoxy groups for covalent binding are located on the outside of a kind of brush structure. This brush structure is, due to its properties, reducing unspecific binding to the plate surface significantly. Of 4 different antibodies on the 3D epoxy surface resulted in a significant amplification of signal intensity in comparison to control plates. To summarize, the 3D epoxy surface allows covalent binding of biomolecules via nucleophilic groups with coating buffers. The surface features a low non-specific background and high chemical and mechanical chemical robustness. 3 microplates are free of detectable DNases and RNases and have a shelf life at room temperature of 2 years. Plates with 3D epoxy surface are available as 96 well black microplates with transparent film bottom and transparent and white F8 strip plates. The last part of this presentation will touch on critical parameters for assay development. We start with introducing a typical procedure for an indirect ELISA. First step is the cutting of the microplate, in this example with an antigen. The blocking step, binding sites still free after coating are blocked to avoid unspecific binding later on in the assay. Commonly used blocking reagents are BA, gelatin, casein, or commercially available blocking reagents. In a step, the sample is added and specific antibody, if present, will bind to the antigen. Where all other molecules not bound are washed away, a labeled secondary antibody is added for detection. After additional washing, a straight for the enzyme label is added, and the resulting signal is measured spectroscopically. A parameter which may have influence on the assay performance is the choice of coating buffer, as the age of the coating buffer has influence on the charge of the molecule to be immobilized. In this example, we immobilized biotinylated insulin on the plate surface. Bio was detected with a Streptavidin horseradish peroxidase conjugate. In example, different coating buffers carbonate at pH 9.6, PBs at pH 7.4, and assayed at pH 5.5 had no impact on the binding curves. The second example, our aim was to immobilize the 10 amino acid strep tag, which is usually used as affinity tag for protein detection. Purification. In that case, the choice of the coating buffer made a significant difference. Occurred only with PBS as coating buffer. In establishing a new assay, optimal working concentration for all components must be determined empirically. This is usually done with a so-called checkerboard screen, where serial dilutions of 2 assay components are screened in a matrix. I will the proceedings using the example of an indirect ELISA based on the immobilization of insulin. In the first step, an appropriate coating concentration is determined. The insulin coating concentration is titrated against the concentration of the insulin antibody. Important in that step is to add the secondary antibody enzyme conjugate in excess to avoid any influence of this component to the assay results. Detection, the optical density values are plotted against the serial dilution of insulin, we're seeing in saturation curves. And saturation occurred, this example, at approximately 800 nanogram per milliliter. Step, the detoting concentration is used, and anti-insulin antibody concentration is screened against the concentration of secondary antibody conjugate. Criteria for suitable combination of concentrations, optical density value of around 1, and the location in the linear part of the curve, as only here slight differences in analyte concentration can be detected reliably. Another important criterion besides signal intensity, suitable signal intensities are here shown in gray, is signal-to-noise ratio. It should be as high as possible. But naturally, it is from an economic point of view no option to use the secondary antibody conjugate undiluted or in very low dilution, which if so with a combination marked red. As the reactions involved in ELISA are kinetic reactions, conditions like incubation temperature, time, and storage conditions play an important role for the performance of the assay. Incubation temperatures range from 4 degrees Celsius to room temperature and sometimes even to 37 degrees Celsius. The impact on the choice of incubation temperature has the stability of the involved molecules. Naturally, the incubation temperature has influence on incubation time. The lower the temperature, the longer the time required to reach an equilibrium. The incubation times range in dependence of temperature from hour to overnight. Another important aspect is the way the microplates are stored during incubation. The best way is to seal them with the sealing tape, to store them under controlled conditions in a unified chamber or a storage system. Stacking of the microplates may result in a phenomenon called edge effect. The effect results from a temperature and evaporation gradient between the inner and the walls of a microplate and is characterized by significant differences in the measured OD values. In stored openly stacked OD values, in standard readings may differ between the inner and the outer walls, like the red curve in this graph. This effect can be avoided by incubating the plates, for example with easy seal, or storing them in a humidified chamber. It's also very important to let all reactions be equilibrated to the incubation temperature before usage to avoid the temperature gradient within the microplate, because temperature adaptation will take place faster in the outer than the inner walls of the microplate. The last point I want to talk about today is buffer composition. Besides substance, commonly a detergent like Twin 20 is added in low concentration to reduce unspecific hydrophobic interactions. Not widely used, but sometimes very important, is the addition of salt to reduce unspecific ionic interactions. When we established a serum-based ELISA in our lab, we started with a very bad signal-to-noise ratio. This has been improved significantly when we added salt in low concentrations to the buffer. Better part of the data presented in this webinar are shown in the corresponding support material on our homepage. And there you will find as well detailed information about ELISA microplates. At the end of the webinar, I want to thank you all for your attention, and over back to Mackenzie.
Wonderful. Thank you very much. Before we end this session, I'd like to thank you all for attending our webinar series and thank Dr. Hanish for such a wonderful presentation. We would like to remind everyone that our webinar series will continue into 2013 with webinars on topics such as the basics of cell culture and the use of human-induced pluripotent stem cells in drug discovery. Recordings of this and all webinars will be available to those who would like them on our U.S. website, us.gbo.com. We will have these up as quickly as possible. Also, if you would like additional information or the literature pieces you've seen here, please email me at mackenzie.ferrone@gbo.com. I have put my email address in the chat feature box if anyone would like to email me for more information. We would also like to invite you to find out more about what's going on at Greiner by visiting our Twitter, Facebook, and LinkedIn sites for information regarding promotions, events, and new products. We'll be announcing the remainder of our 2013 webinar schedule if you'd like to participate. So let's jump into some questions. First, is there a guideline for specific addition to enhance signal-to-noise ratio?
For starting, I would try sodium chloride, magnesium chloride, or potassium chloride in an isotonic concentration range.
Okay. Are color-coded strip plates available to facilitate the discrimination of different applications?
Color-coded plates are available in the single break strip format, I think in red, blue, green, and yellow color codes. F8 striplets are unfortunately not available color-coded.
And we have another question here. With CV of inter and intraplates that you had mentioned being somewhere between 5 and 10%, what kind of CV may one expect of the full product one may either produce in-house or in the commercial products?
This really depends on depends on the detection method. As luminescence or fluorescence detection is much more sensitive than colorimetric detection, the CV has to be higher, 10% instead of 5% for the colorimetric assays. For the product, we cannot make any suggestions because this depends on a company which will use plates to produce this final product.
Okay. What is the code area for Streptavidin coated microplates?
For 96 plates, it is 300 microliters, and for 384 plates, it is 90 microliters.
Okay. An additional coupling step needed to attach a protein to the 3D epoxy surface?
With this surface, no additional coupling step needed for covalent binding. You can use your standard coating buffer.
Okay. And those are all the questions that have been typed through the chat feature. And I did put my email address in the chat box, mackenzie.furman@gbo.com. If you have follow-up questions, if you'd like a copy of the recording of this webinar or any of the literature pieces Dr. Hanish talked about today, we'd be happy to provide that to you. If there aren't any further questions, I'd like to thank you again for attending, and thanks again to Dr. Hanish, and we hope that you have a wonderful day.